All right, let's get started. Thank you for everyone for attending our webinar on what is a taxonomy. It's a part of our series of what cell types, taxonomies, A to Z's. My name is Lauren Filer. I'm part of the Education Engagement Program, and I'm just going to be your moderator today and take care of some housekeeping rules for today. So reminder, this webinar is being recorded. If you're wanting to see this recording, it will be posted on YouTube after the webinar is over. If you'd like to attend any more webinars in our series, please register for all future webinars. Links will be sent out. We're happy to take questions, so if you're on Zoom, feel free to post them on chat. If you're on YouTube, you can also post on the YouTube chat. If you're watching a recording of them, we'd love to take questions found in our community forum, which is found in the YouTube description. So for today's agenda, we'll have a video from our scientist, Nelson Johansson, on just building a simplified version of taxonomy. He's on paternity leave, so congrats to him and his family, but he did leave us a great video before he left. What he's going to talk about is a lot of the fundamentals that are going to take over for the next part of the latter half of today. So we really encourage any questions or comments you have or clarifications on what you said on the video. And then our scientist, Rachel Hostler, will talk about how a taxonomy is used in a real paper with walking through the primary motor cortex. And then Kyle will talk about how we apply a taxonomy in our Alzheimer's work. Afterwards, happy to take questions on anything Rachel, Kyle, or even what Nelson said. And with that, we'll kick it off to Nelson.
Hi everyone, I'm Nelson Johansson, a scientist at the Allen Institute for Brain Science, and I'm really interested in evolution and how brains and cell types evolve through species such as mouse and primates. So today I'm going to talk to you about what is a taxonomy. At the Allen Institute, we build taxonomies based on the RNA or gene expression of a cell. And in my mind, a taxonomy is just a way to organize cell types into higher order groupings that have some sort of relationship between all those cell types. So in order to build a taxonomy, our foundational component is single cell RNA sequencing. So in order to perform single cell RNA sequencing, you of course take your tissue, dissociate it, and then capture these individual cells in droplets. This allows us to get to this single cell resolution of RNA or gene expression. So once you have these cells in droplets, you take those individual droplets, lyse the cell, and capture all of the different RNA fragments that are present in that cell at that time point. And from the RNA, you reverse transcribe it into cDNA and pass that through what's known as a sequencer. So the sequencer takes those fragments and measures what they are and captures how many of them exist for a single cell. And this is really the workhorse of a lot of our taxonomy building, is isolating millions and millions of cells, sequencing them, and then forming what's called a cell by gene matrix, where this matrix has all the cells that we pass through the sequencer and all the genes whose expression that we measured during this process. And this ends up building these matrices that can be millions of cells wide with around 30,000 genes. So once we have this cell by gene matrix, that really is the building block for the taxonomy. And what we're interested in now is how you take all these cells with their different gene expression profiles. Some cells highly express certain genes, other cells highly express other genes. And that kind of is what defines a cell type, the expression pattern of marker genes that is unique to that set of cells. So in order to organize cells into groups, we use algorithms called clustering algorithms. And this is the next step in our process towards a cell type taxonomy. So here, we're going to take those millions and millions of cells and cluster them. And the core idea here is that I'm going to draw a few cells as circles, and we try to understand how they relate to each other in terms of gene expression. So finding links between cells that are highly similar based on Euclidean distance or correlation. And then from there, we of course have many more cells, and finding how those relate to others, again, based on their gene expression patterns. And doing this until we've built all these beautiful communities of cells that organize based on how or what genes they express. And the nice analogy here is that you have cluster one, for example. This is your group of friends on Facebook and how you all interconnect. And in another group of friends, cluster two, might have a common friend with your group, but also have their own, friend community. So the idea with clustering is that we're just taking all these cells and finding these communities that have common expression of genes in their expression profiles. So once we have these clusters, the next step is starting this process of taxonomy building, which I'll show you over here. So with our clusters or communities of cells defined, we have the foundation for our taxonomy. And here I'm showing you 6 clusters that we're going to build into well-defined cell types throughout this talk. So the next step is, once you have clusters, is identifying low-quality data and making sure that you remove that so you only have high-quality expression building your taxonomy going forward. I'll talk about that next. All right, so now we're going to talk about quality control, and that is identifying cells and clusters of cells that have a different pattern of gene expression than what we'd expect. And this pattern can fall into a few categories. One of the first one being just low gene expression. So we expect that cells will express a certain number of genes if the sequencing and all the technology there did a good job in capturing the RNA fragments. So if a cell has, 10 to 100 genes in our tissue, in the brain, that is really, really low. And that identifies cells as being of lower quality. And why this can happen is a few cases. So one of which being just the reagents. So the reagents used during the sequencing process can influence the number of RNA transcripts detected. A second reason being is that the cells could be going through cell death. And that is, they're losing, or their RNA is degrading, and we can't capture the full space of gene expression that we would want to for this individual cell. So low gene expression is the first kind of indicator that a cell is low quality. Another indicator also kind of comes from this technology side, where we can have what are called doublets, or basically multiple cells in the same droplet, like I showed previously. And this leads to RNA mixing between cell types that can really confuse taxonomy building, because you're no longer capturing a single cell in these expression measurements. These are 2 really nice indicators that we've found at the Allen Institute. We track all this information from the cell going into the sequencer, the reagents used, who did it, time of day, and that is all really useful in figuring out what are the low quality data in our data sets. So going from there, it's also important to make sure that you're not throwing away good cells. Certain tissues like spinal cord have these really massive motor neurons that express tens of thousands of genes. And sometimes if you set your thresholds wrong on what is low quality, that can get picked up. So this is a very critical step. It's easy to make mistakes. So we're always very careful that we understand our tissue and our kind of range of interest and make sure that we know what kind of types should be there to figure out how to identify low quality data. So once we have the quality control per cell, we can go back to our communities and find cells that have low quality and flag them. And as you can see, this community here has 2 cells that are low quality. For us, if there is high enough proportion of low quality cells in the community, we throw the whole cluster away. This cell may have just barely been good enough to pass our quality control filters, but was still insufficient compared to the other high quality data. So we throw the whole community away going forward. And now I'll show you that on our taxonomy. So now we know which clusters don't pass our quality control filters, and we can remove them from the taxonomy and don't include them in any of the further steps. So now we're going to talk about how to actually merge clusters into higher order groupings with supertyping. So now that we've defined our clusters and which ones are high quality, the next step is to actually merge those clusters into cell types. And our first step towards that goal is by doing an approach called supertyping. And the idea here is we want to find clusters that are very well identifiable. That means they have very clear marker genes. Genes that are individually or uniquely expressed in that cluster. So very clear expression of these marker genes. And the way that we identify what is clear or robust expression of marker genes is by training what are called classifier algorithms on our single cell data, on our clusters, and asking the question, which clusters can we classify well? That is, which clusters have really good marker genes for their expression? And we do this for all clusters, and we'll find certain clusters that, well, the model just can't figure out. And that could be because the clusters define more gradients in expression, and we want to be able to capture that in our taxonomy. So I'm going to show you what this looks like on our taxonomy. So in this example here, our classifier will have been trained across all of these clusters, and it'll tell us something like, I can identify this cell type half the time, along with cell type 2, or cluster 2. So supertyping tells us that, okay, we're not happy with this accuracy in classification, so I'm going to merge cluster 1 and cluster 2 into a higher order supertype. And once we've merged these two clusters, now we retrain the classifier and ask the question, how well can you classify supertype 1, cluster 3, 4, and 6? And if we're happy with this accuracy, we can then go ahead and say we're happy with this grouping of clusters and to move forward with our taxonomy building process. And just to be clear, in this example, the other clusters we could classify well. There was good marker gene expression. So this is the first step towards our hierarchy or our taxonomy of cell types. The next step being taking known literature-based marker gene expression from studies that have been going on for decades to define what are the genes that are expressed in various neurons and non-neurons of the brain. So this is taking literature, all of the body of work from before our study, and incorporating that into the taxonomy. And how this works is we'll go back to our taxonomy tree here, and we're going to start asking the question, for known marker genes of inhibitory neurons, say SST inhibitory neurons, which clusters express SST? And in this example, we can imagine the first 3 clusters express this SST marker gene. If that's the case, we can then merge them together into this SST subclass. So now we have a new definition, which is just a higher order grouping of the previous 2 levels of our taxonomy. And this grouping is completely based on, again, the expression of known or de novo or new marker genes that we find in our data. This defines the SST subclass, but then we know that above SST there are these inhibitory neurons, a wider collection of them, and they express marker genes like GAD1. And you can imagine that clusters 1 through 4 all express GAD1 along with their lower level or subclass level marker gene expression. If this is the case, we would then merge all these inhibitory neurons into what we define as a class. This is a really broad definition that defines the major types of neurons or other types of cells in our tissue. And again, this is driven by going to literature, understanding what people have looked at in the brain, and utilizing that to inform our taxonomy. And of course, this is easy to draw here, but this takes a lot of time in building a taxonomy. You have to know what genes to look at, how to group clusters, and making sure that you have the right marker gene sets to define your grouping of clusters. So once you have this inhibitory class, then of course you have other types like excitatory neurons, non-neurons. I'm not going to show that today, but the logic is the same, where you would go into all the clusters of excitatory neurons, find known marker genes at the subclass level and the class level. And then you connect them all up with our taxonomy as another branch. Then you have multiple branches that define classes and higher order groupings of clusters for the whole tissue. So with that, we've gone from a very high resolution clustering of data. We've merged those into supertypes that can be very well identified from gene expression profiles. We've used literature to tell us what are known types in our tissue at the subclass and the class level, and built this organization, this taxonomy of clusters so that we can communicate what the landscape of cell types looks like in our tissue or brain region of interest. And the goal here is to be able to share this with the community so that they can take our understanding of cell types and use those for studies of disease, of evolution, of any interest in this region of this taxonomy is being built on. So with the taxonomy now fully defined, we share these with the broader neuroscience community so that they can relate their research to this hierarchy of cell types that we've defined.
Thanks again, Nelson, and we wish him best of love for his and his family for that. And so here's just a quick recap of what just Nelson talked about for the taxonomy. And so then it's a simplified version again, as you said. You use the single cell RNA sequencing to make clusters. And then we use the classifier to, we use quality control to remove any failed cluster. And then we use a classifier to merge the clusters for a supertype. And then we use marker genes with our literature to merge for subclass and class level. And then we connect all the other classes and clusters to complete the taxonomy. And with that, welcome any questions. We already got a couple in the chat, but please, if you have any questions on what Nelson said, there's no stupid question, any fundamental, this is what we're here for. So the first question is the supertyping classification one versus all other clusters or one versus one? Example, pair of clusters. And I think, Kyle, you want to take that?
Yes, I can answer that. So we typically do supertyping within the set of clusters that are highly similar to one another. And so that starts with, one versus one kind of pairs of clusters. But then as we're building the taxonomy, it's sort of an iterative process where if you define a subclass, you then want to make sure the clusters are robust against all the other types within that subclass. And then there's the final high level of kind of one versus all. But by that point, things are fairly robust. What we don't do is one versus all without any consideration of the hierarchy, just because in that kind of process, it can be challenging to distinguish two highly related clusters when you're also distinguishing further related clusters. So hopefully that was clear.
And here's two questions. We'll do the first question. How do we check the correctness of a resulting cluster? Of course, clustering is an unsupervised learning algorithm, so we do not have the true answer, but is there any checks that we can perform on the clusters?
So the way that we do that supertyping procedure is if we've created a taxonomy based off of, say, 5 donors or 6 donors, we'll use a hold one out approach where we will remove a donor sequentially and train on the remaining donors, and then try and see how well we can predict the types in the held out donor. And you'll do that holding out each donor iteratively such that you have a prediction kind of for every single cell type. And that's, a common bootstrapping procedure basically to ask, can I recover that original label with a subset of the information? So is it kind of robust to that subsetting procedure? And so I would recommend those kind of things in kind of interrogating and challenging your clustering.
And the second part is, do you define the number of clusters in advance, or using a clustering algorithm to decide which number of clusters based off the data?
The clusters are defined kind of hierarchically in a process where you oversplit and then pairwise merge all of that. And so we're not saying the number of clusters in advance. In fact, we're trying to create kind of very small fragments of cells at the outset. And then we actually do pairwise differential expression testing to see, okay, how many marker genes are actually different between these two kind of really small fine grain clusters, and then merge them together if there aren't enough differences in the creation of those clusters up ahead. And that's a lot of that logic is implemented in the Scratch HiCat package that Alan has put out.
Can supertype and subclass/class classification overlap?
So I'm not sure what the question is in terms of overlap. Generally speaking, every single supertype has one subclass, and then every single subclass has one class. So there's not degeneracy in that way, if that's what they're asking.
Then I guess a further clarification, could there just be one supertype that equals one subclass?
Yes, we do have examples where you can no longer further split cells, and so we just keep propagating those cells as a subclass and then as a supertype and then as a cluster, just to have all of the levels of the dendrogram present. But in reality, the subclass would be equivalent to the supertype would be equivalent to the cluster.
All right. How would you determine the connections of neurons in subtypes of glia? I've seen trees that split neurons in glia first, or say split microglia versus other cell types first.
Connections of, yeah, so we in our taxonomy creation, almost always the first level split is inhibitory neurons, excitatory neurons, and then glial cells, so astrocytes, oligos, and OPCs. And then the final one is non-neural cells, so microglia, endothelial cells, fibroblasts, ependymal cells, things like that, that don't have a lineage relationship. So it's really that kind of split, and that's really inspired by developmental origin and similarity.
I have another question going back about the hold one out approach. What model do you use to train the hold one out approach? You mentioned that you trained something first.
Yes. So for those hold one out procedures, we're using a combination of SCVI and then ScanVI. So these are based off variational autoencoders that compute a joint latent representation. And then if you have some data that has labels and some data that doesn't have labels, you can ScanVI uses a classifier to basically predict labels for the data that doesn't. And then when we are thinking about multimodal data where it's not just single cell RNA-seq or single nucleus RNA-seq and we have attack, there are tools in that universe, such as multi-VI, which do a very similar thing. So those are our workhorse algorithms currently.
What? We don't have any more questions right now, but if anyone has any more questions, we do have another Q&A session after Rachel and Kyle speak. Feel free to drop them in the chat now or save them towards the end. Either works. And with that, I'll take it over to Rachel.
All right. Hi, everyone. I'm Rachel. If you watched our first webinar, you may recognize me. It's nice to see everyone again, and welcome to part 2 of our webinar series. So I'm just going to skip through a couple of these slides since they just summarize what Nelson had talked about. And now I'm going to be talking about a couple examples of taxonomies generated by the Allen Institute and their collaborators specifically focused on the primary motor cortex. So in the example that Nelson just gave, that was a very basic example showing what happens when we maybe have 6 or 7 clusters of cells. But in reality, things are much more complicated when we have many more cells, many more clusters in a larger brain area. And so this question was tackled by the Brain Initiative Cell Census Network, or BICCN, in 2021, where they created a mini atlas of the primary motor cortex in 3 different species. So this involved collecting tissue from mouse, non-human primate, and human, and then trying to create an atlas of all the cells, specifically in the primary motor cortex. And why primary motor cortex? It's an area that's highly conserved between species and has a lot of previous literature. And so those are just a few reasons why that was selected as the first area. And as some of you may know, just last year in 2023, BICCN, or now BICAN, the Brain Initiative Cell Atlas Network, created a whole mouse taxonomy. But now this is just focusing on primary motor cortex. And so the result of this project focusing on primary motor cortex resulted in 17 publications, including 1 flagship publication that summarized all of this work. Over 200 terabytes of data were generated. And to help sort through this data and to help it make it more accessible, BICCN created the Cell Type Knowledge Explorer, which is an online interactive open resource to access the data. And you can find that at brainmap.org. And we'll be talking more about the Cell Type Knowledge Explorer specifically in our next webinars on February 28th and March 27th. All right, so in Nelson's example, again, we had 6 or 7 clusters of cells. From data from the primary mouse motor cortex, we ended up with 116 clusters, which ended up being 116 cell types, which then are nested underneath 24 cell subclasses, which are then within 4 cell classes. And so here's what that looks like. This is a dendrogram showing all of the cells that were analyzed and characterized in the mouse primary motor cortex. And you can see here we have the split at the beginning between our non-neuronal cells and our neuronal. Then once we have our neuronal cells, we have our split between our GABAergic and glutamatergic. Then we have further splits based on embryonic origin and other differences. And again, this is all based on transcriptomics. So this dendrogram specifically was created from multiple data sets of single cell and single nucleus RNA-seq data, and also some ATAC-seq and other omics data as well. And so that's one way to show hierarchy and to show a taxonomy, is through this display here called a dendrogram. Another way is through a sunburst plot. So the sunburst plot here on the right is showing the same exact data here on the dendrogram, but it's just showing it in a different way. So here in the sunburst plot, this is also showing hierarchical relationships. And what we can see here is that on our inner ring, we have our 4 main classes. We have our GABAergic class, our glutamatergic. We have 2 non-neuronal classes. And then once we split further down, we are divided into our subclasses. So our GABAergic, we have somatostatin, we have parvalbumin, and then further down, we have more splits as well, looking at our individual subtypes. So these are just 2 ways to visualize taxonomies. And here's an example of the mouse primary motor cortex. And so, as I mentioned in the previous slides, this was actually done on 3 different species. So I've talked to you a little bit about our mouse taxonomy, but at the same time, there were data sets created and generated on marmoset and also human. And when you look at these 3 different dendrograms, at first glance, they look very similar, but there's also details and differences between them. They're not exactly a 1-to-1 ratio between human, marmoset, and mouse. And so to really compare and see where we're very similar in these species and where we're very different, the BICCN researchers then integrated these single nucleus RNA-seq data sets together to create one integrated taxonomy, which is shown here. And in this image on the right, these blocks down here show the different cell types. So right here, all the way on the left, if we look at LAMP 5.1, if we specifically look at that cell type group, we have one of those in human, one in marmoset, and one in mouse. So that is a one-to-one relationship between the 3 species. However, if we look at some other cell types here, you can see that, especially in the part-valve human cells here, the P-valve group 1, there's lots of different cell types in each of the groups, and not every cell has a one-to-one relationship. And also, if you look in the dendrogram up here, it's actually color-coded, if you can see, between sort of red and black, looking at the confidence between the different links as well. So by combining these data sets together and integrating them into one taxonomy, we can really see where are we extremely similar between our 3 species and where are we different. And this is just, I'm just briefly discussing this cross-species taxonomy, but we'll actually go much more in depth into cross-species research and cross-species taxonomies, specifically on March 27th. And so all the research data that I just showed you was specifically based on transcriptomic data, but there were many other experiments that were done on these samples from our different species, specifically looking at spatial data, so looking at the spatial transcriptomics, experiments to look at the electrophysiological properties of cells, or to look at the projections, the full morphology or the local morphology, and also a lot of different transgenic mouse lines were used as well. And so these experiments were really mapped onto the backbone, which was our transcriptomic taxonomy. And so here again is specifically just the mouse transcriptomic taxonomy. But then there were also other experiments done with PatchSeq. And I briefly mentioned PatchSeq in the previous webinar. PatchSeq is a technique where you can record the electrophysiological properties from a cell, fill it with biocytin, and at the same time also extract the nucleus and do sequencing on the nucleus, so that from the individual cell, you have a combination of transcriptomic data, morphological data, and also electrophysiological data. And PatchSeq was performed on these different cell types. And just from looking at their firing patterns and their electrophysiology, researchers were able to identify them and link them and map them onto our transcriptomic taxonomy, which gives us a better idea of these cell types by combining different types of data modalities. Again, with PatchSeq, you also fill the cell with biocytin, so then you can reconstruct its morphology later. And those experiments were done as well. So not only can we see how these cells differ transcriptomically, we can also see their unique firing and their unique shapes. And this is one way that these different data modalities, which are very important, we're able to map them onto our transcriptomic backbone. Here's another example of this. This is specifically looking at the long-range projections of our excitatory cells. So here are some examples of layer 2, 3, layer 4, and layer 5 IT cells and layer 5 ET cells. And so our IT cells are intratelencephalic, meaning they keep their projections within the telencephalon. You can see here that, again, these are all from the primary motor cortex. So they're projecting from the primary motor cortex to other areas. And we can clearly see in our IT cells that they stay within the telencephalon, and our ET cells project to different areas outside of the telencephalon. And so, again, by doing these other experiments on top of our transcriptomic experiments, we now have a much richer and much fuller picture of the identities of these cells, because we know that not all IT cells project to the same area and not all ET cells project to the same area. So by doing these additional experiments, we can really characterize these cells further. And so those are just some examples of some of the recent taxonomies generated at the Allen Institute and with our collaborators. And every example that I showed so far was just research and data from healthy animals. So healthy human tissue, healthy mouse, healthy marmoset. And in our next segment, we're going to move on to Kyle, and he's also going to talk about taxonomies, but he's specifically going to talk about how taxonomies are affected by different disease states.
Thank you so much, Rachel. So I'm going to talk about our work on the Seattle Alzheimer's Disease Cell Atlas Project, or CAD, because it really highlights why these taxonomies are so important, even beyond sort of basic science research, but for the research into different diseases. So in CAD, we are collecting data from human donors that have died with various degrees of Alzheimer's disease pathology. So some of our donors are totally cognitively normal and have no AD pathology whatsoever. And then we have donors that have sort of mixed levels of pathology, maybe slight mild cognitive impairment. And then we also have donors that have really high levels of plaques and tangles inside of their brains and frank dementia. So really the full spectrum of the disease. And across 84 donors that we have so far, we've profiled millions of cells from several regions of the cortex. And what we could do with this data is create another taxonomy following the exact same approach that we just described here. We could take all of these cells, cluster them based off of their similarity, and then create a super typing by looking at how similar and resolvable those clusters are, and then use marker genes to build subclasses and classes. But that would be repeating a lot of work that had already been done. And then at the end of the day, we would still have questions of how all of the types that we defined in our CAD data set related to the ones in the reference data set. And so instead, we want to map these millions of cells onto the 100 to 150 types that we know are in the cortex that are organized at the class, subclass, and then cluster level inside of the human cortex. And so just coming back to Nelson's example, what this looks like is we have a new cell, and it's asking a question, which is, what am I? What kind of cell is it? And in order to do that, you look at the genes that it has expressed and how similar they are to, first at the class level, inhibitory neurons, excitatory neurons, or some of the non-neuronal populations. And let's say it looks a lot like an inhibitory interneuron based off its expression pattern. It would then move down into the hierarchy, and then you would ask, okay, what kind of inhibitory interneuron am I? Am I a somatostatin positive, a parvalbumin positive, or maybe a other VIP, SCNG, LAMP5 positive population? And if it looks a lot like an SST interneuron, then it would get mapped into that at the subclass level. And then once it has a subclass, within that subclass, we then ask the question, what supertype or mappable cell type does it resemble and does it look like? And I just want to say, in this process, this uses a deep generative probabilistic algorithm that I talked about and described kind of in the Q&A session. And we've made a web-based tool where people can go and upload their own data and then map using this approach to the CAD taxonomy online. And what this will return, if you upload your own data, is a list of all of the cells within your data set, and then a prediction of the label, and then a probability that comes from the model for how confident we are in that label. And the map my cells is also going to get talked about more in depth at a later webinar. And the reason why both the label but then the probability are important as we walk through sort of this cell type mapping hierarchically is that very often when you're mapping disease data in particular to a reference that was defined on healthy tissue, you might encounter cell types that don't really look a lot like any of the others that are in the reference. And actually, in Alzheimer's disease, we have a very prominent example of that where while normal microglia have a very thin morphology, in Alzheimer's disease, microglia can actually adopt a very activated or inflammatory state, which results in morphological changes in microglia themselves. And so we had to identify these cells based off of them having low probabilities in mapping in our model and not really looking like anything else that we had, and then add them to our taxonomy. Other examples of things that could cause something to not be in your reference to look out for as you're mapping is There could have been a rare population that wasn't captured in the reference, but is in your data set because you've more deeply sequenced a region. There could be populations that are only present developmentally because they're precursor cells that are not seen in an adult context, or it's a state that is only observed in an extremely aged individual. Or perhaps a cell type that is only captured with certain specialized techniques. Say a cell has a lot of lipid droplets that are present inside of it, and it kind of floats away for the most part in a lot of preparations, but you have specifically targeted that population to capture it. And so those are just things to look out for as you're mapping, is to not take those mapping results at face value, to really consider the probabilities if you're using a probabilistic model like we have in Map My Cells, and to use your biological intuition for whether or not things are making sense, or if potentially you have a population that's not in the reference. If we do have a reference population, or if we do have a population that's not in the reference, what we typically do is we just add it to our taxonomy. And what that looks like in the context of CAD, I'm showing you here in gray all of the nuclei from CAD donors, and then colored nuclei are those that came from the reference. We're using the integration and then ScanVI to make predictions for what they are. And if you zoom in on the microglia, you can see what the original predictions from ScanVI were, and then how we've expanded that taxonomy. And the reason we've done that is in areas of the graph, there were just not a lot of reference cells. So these arms right here, you can actually see two new populations that did not have a lot of reference cells in that part of the nearest neighbor graph. And so we added them to our taxonomy using a clustering based approach like the one I described. And so the result is, for the CAD taxonomy, we have our class level labels: inhibitory neurons, excitatory neurons, and then non-neuronals, the 24 subclasses that are kind of common across the cortex, and then 139 supertypes. The reason why mapping to the reference is so powerful is we can then ask questions like, what are neurons are specifically vulnerable in disease? So which, as a function of disease, do we preferentially lose? And what was really interesting is in CAD, we found neurons that we kind of expected. So these are the neurons that get the neurofibrillary tangles, the excitatory IT neurons in layers 2 and 3, and to some extent in layer 5. But we also saw several inhibitory interneuron populations that were being selectively lost, and glial changes, some of which were expected. But we could also then look at not what was being lost, but when, as a function of pathology, these changes were occurring. And what was really interesting to us is you had a huge exponential ramp in the number of plaques and tangles at a certain point of sort of that trajectory. And that's roughly when you were losing these layer 2-3 excitatory neurons and the parvalbumin interneurons. But really notably, the somatostatin neurons were being lost much earlier and kind of before there was a huge amount of plaque and tangle pathology in these donors. And what having the reference mapping, so knowing what each of these populations were, allowed us to do was then to look at what are the morphologies of the cells that are vulnerable in neurotypical reference. And that lets us see kind of what are the types. So we have non-Martinati cells, sparse SST, but also the human-specific double bouquet cells were also present. All of them were in supergranular layers, so they were all in those superficial layers that were next to the layer 2-3 IT populations that eventually were lost. But then we could also look at electrophysiological properties that were unique to them as well. And one kind of notable difference between the vulnerable somatostatin positive neurons and the unaffected somatostatin positive neurons was after spiking, the vulnerable neurons had significantly higher SAG. So they depolarized further than the unaffected types on average. And so this just gave us important context for not only naming transcriptionally what cells were vulnerable, but really having a good idea for where these cells are, what they look like, and then what their ephys properties are, so that if anyone wanted to study these cells in the context of Alzheimer's disease, they would have so many different ways of identifying them and accessing them overall. And with that, I just want to thank you and pass it back to Lauren.
Thank you, Kyle and Rachel. Oh, Kyle, can you go back
Sorry.
to screen?
My bad.
You're all good. All good, all good. But yeah, thank you, Kyle and Rachel, for that. And we'll be about to take questions. But just a reminder, if you want to hear any of the webinars that were called out, such as across species, how to map it, you have to register for each webinar. We want to make these webinars better and better. So it really helps us if you guys take the survey. It's a quick less than 2-minute survey of what ways we can make our webinars better. And with that, we got a couple questions in the chat, and I will answer them once. So question one is, after I upload my data, can I receive more than, I'm guessing this is about map my cells. So after I upload my data, can I receive one of the mapping results as an unknown class, or will I always get a result with one or existing classes, both with low probability?
So the way that Map My Cells works will always give you a prediction, but if, let's say, a cell was often QQQ and looked nothing like any other cell, we would expect that the probability of that prediction would be lower than the probability of predictions for cell types that are, more highly confidently mapped.
And then another map my cell question, can map my cells be applied to brain regions other than MTG?
So we've looked carefully at how the MTG relates to another cortical region, the dorsolateral prefrontal cortex. And those types are so similar that they're essentially one-to-one. And so I would not have kind of any qualms about mapping data from that region. There are cortical regions such as V1 that have populations that are unique to that cortical region and look kind of different. And so there, I would trust the mapping down to the subclass level, but maybe not the specific supertype or cell type overall. But broadly across the neocortex, a lot of the types are very similar. In fact, there's a paper in the package that Rachel mentioned that looks at similarity of types across the neocortex. And I will say we are working very actively on tools that then will allow mapping to subcortical structures like the basal ganglia, hippocampal formation, and then other brain regions as well.
And again, that webinar is April 24th. So if you register for that, you'll hear some of the scientists who help develop it and get more direct answers and can ask a lot more technical questions there as well.
And I also want to mention that so when Kyle was talking about Map My Cells, he was specifically talking about the human Alzheimer's data, but we also have a data set for the whole mouse brain. So if you're doing mouse research, we have the entire mouse brain up there.
No more questions. Leave it up for a couple more minutes. Otherwise, let people have 10 minutes back of their day. If any other questions come back up, we always have a community forum, which is answered by our scientists. So they're happy to take questions there. Recording this will be on YouTube. So if you miss something, it will be there. Links will be sent out to the same for people who registered. But if not, it's on our Allen Institute channel. I think with that, we can call it a day. Thank you, Kyle and Rachel, for all their help today. And thank you for everyone.